In this video, we will wrap up our Hardy-Weinberg chapter. We're going to be looking at conditional probability and how that's associated with DNA and how we have used probability for DNA calculations. Now, conditional probability is the measure of the probability of an event occurring given that another event has happened. So, for example, in the F2 generation of a monohybrid plant, where we have tall plants and short plants, what would be the probability that a tall plant is heterozygous? The condition that we set is to consider only tall F2 offsprings, since we know that all the dwarfs are going to be little t. So of the F2 tall plants, what is the probability of it being heterozygous? That's an example of a conditional probability. Doing conditional probability, many times we'll use pedigrees and Punnett squares. We'll use Mendel's law to predict the occurrence. Let's look at an example. Tainshina's brother, Deshaun, has sickle cell anemia. This is an autosomal recessive disease. What is the probability that Tashina's child will inherit the sickle cell anemia allele from her? So because the outcome of the conditions are not independent, we cannot apply the product law, so we'll have to use conditional probability. So right here we have the pedigree of the family with the parents and the offsprings, and we can see that Deshaun is homozygous recessive and has the condition. Now, if we take the parents and we do a Punnett square and we take this area off, because we know that Tainshia is not sickle cell anemia, that's gone. And we look, she could be a carrier here and here. So 2/3 is the probability of her being a carrier. Now the probability that the child will inherit the sickle cell allele is 1/2, because if she is this right here, she would have a 50% chance of receiving this, or a child would have a 50% chance of receiving that. So the probability that the child carries the sickle cell trait would be the 2/3 times 1/2, which would be 1/3. Now we're going to combine our DNA information on this particular video as well. So when we look at DNA repeats, we're looking at short repeated segments of DNA over a genome. And the repeat numbers can be considered alleles, and they can be used to classify individuals. So we have two types of repeats. We have the short tandem repeats. We're going to abbreviate that STRs. And we have our variable numbers of tandems, and we're going to abbreviate that VNTRs. So when we look at our STRs, which are small, that's 2 to 10 bases. When we look at our VNTRs, which are much larger, that's 10 to 80 bases. These are our fragment sizes, and so here we could have some examples right there. So that we want to find out as much information as we can in order to do DNA profiling. This is the technique that detects differences in repeat copies. It allows us to calculate those probabilities, such as conditional probability, the Hardy-Weinberg, and all these other probabilities that we've been doing on previous videos and homework, so that we can use DNA evidence, and that can be more valuable from excluding suspects as it can be to determining who is guilty. DNA profiling was first developed in the 1980s by the British geneticist Sir Alec Jeffreys. I believe that's a test question. It's also called DNA fingerprinting. It helps us to identify individuals. It can be used in forensics, agriculture. It can be used in paternity testing. It can be used in historical investigations. So that what happens is that depending on those sizes of DNA, the smaller the DNA is, the smaller the fragment, it's going to move faster And then the larger the fragment is, it's going to move slower. So when we use a gel and we do electrophoresis, what we do is we look at the repeats, and we'll use an enzyme. We're going to cut the DNA at very specific places. It's going to make these small fragments. We put it in the gel, we run electricity through it, and then it's going to make these fragments. And again, smaller fragments move faster, and larger fragments will move slower. So that here you can see what one of these DNA electrophoresis gels looks like. And so we've got it marked at how fast or how far they've moved. And they move from the base area. And so each one of those is a line. And you can see that we're matching up lots and lots of lines. And those are all DNA fragments. And the more fragments you can match up, the closer that you have that those individuals are connected and that no other individual can match up. So Jeffrey used his technique here to demonstrate Dolly was truly a clone of the sheep that donated her nucleus. Now, to do the DNA profiling, you need a blood sample. You can use cheek cells. That's becoming very popular. You do electrophoresis, and then you look at the pattern of the DNA fragments. And those fragments will show up because they're exposed to a radioactive probe. The probe binds to the DNA, and then you can use X-ray film and you can take actual photographs. This used to be such a lab that it was done when I was getting my master's degree, and we would spend somewhere in the neighborhood of 30 hours in a week doing one lab. Today, this is such a common procedure that you can do this in a high school lab. So that you can get the DNA from either the blood from either suspect. You can then use SNPs, enzymes to cut the DNA in very specific places. Then you're going to put those DNA samples into the gel. You're going to put the gel in the electrophoresis. You're going to run electricity through it. The gel is going to separate out into bands, and then you could take the photograph and then you match them up. So in this example right here, you've got, so we'll say, okay, here is the crime scene. Here are my two suspects. This suspect lines up with the crime scene. The DNA can be obtained from any cell with a nucleus, and if it's extremely damaged, you could use the mitochondrial DNA. The mitochondrial DNA was what was used to identify the Romanovs when they found skeletal remains in Russia, and they were able to identify Anastasia. For forensic purposes, the FBI actually has a combined DNA index system. It's called a CODIS, and it uses 13 STRs. The more STRs that match, the less likely anyone else could possibly be the suspect. Recent examples of using DNA profiles, the World Trade Center attack in 2001, when they've had the different tsunamis and they've had to match up bodies, Hurricane Katrina, it goes on and on. Anytime where you have a mass group of individuals and you need to match them up, then you can get the DNA from hairbrushes, from toothbrushes, from family members, and then you can match them and identify the bodies. Some of the challenges of DNA profiling would be in mass disasters. Mass disasters, it helps the climate causes the body to decay faster, which means that you may not get quality DNA. You may not have a laboratory set up because of that mass disaster. You may have so many casualties. To this day, they are still trying to identify some of the bodies from the 9/11. Now, they're not whole bodies, they're fragments, but they're still trying to identify them. So you could have destruction of personal items, you could have lack of DNA, You could have poor DNA. All of this would cause a challenge, and all of this has caused challenge with identifying the 9/11 victims. Because genetics is such a hot topic right now, today's population of genetics presents really powerful ways to identify individuals. Our genomes can vary in more ways than there are people in the world. And so when they match up, the likelihood of it being someone else is very slim.